Okay, Pirates, welcome to my afternoon viewing of this Chapter 11 Gene Action Study Guide. When we're talking about gene action, we are talking about how DNA works, what it is, and how DNA makes protein in the body. So we're going to be talking about a couple of main things. We're going to be talking about what DNA is. We're going to be talking about this, what it looks like, how it acts, how it works, and we'll be taking that step by step. All right, so what I want you guys to do is if you do not have this all the way done yet, go ahead and follow along. That's fine. What I would like you to do at the end, though, is to retry this all on your own, especially since our test is like 5 days out. So it'd be a good idea to do this a little bit closer to the test. So go ahead and write down the information, use the pause frequently, rewind, fast forward, look for the questions you need. I'll be doing this in order, and I'll try to circle the question I'm currently talking about. Okay, the first section is DNA basics, and DNA basics is just basically what is DNA. So number one, DNA is a code. How is it a code? Well, the code is that the order or the sequence of the bases, remember order and sequence means the same thing, they tell the order or the sequence of a protein. So all a code is a language or a bunch of information, and so therefore the order in the bases is that information it codes for. It translates into an amino acid. All right, number two is important. DNA is similar and different amongst all organisms. Well, first of all organisms have DNA. Every living thing that we are aware of right now have DNA, every single one. Okay? I mean, it might be in the future that we find an organism on a different planet or maybe even on our planet that doesn't have it, but so far every living and even viruses have DNA or mRNA. All right, the main similarities of them are the same structure, so they're all made of phosphate sugars in the same four bases. They have the same function. They make mRNA, and that mRNA goes on to make proteins. The thing that makes them different is the different order of the bases. The different order of the bases leads to different proteins, and those different proteins lead to the organism looking, acting, behaving differently. And so that's where all of our differences come from, all of the variations within a species and all the variations between the species. All right, the two most important functions of DNA is it is the hereditary material or the genetic material. As far as we know, all living organisms eventually will break down, and because they will break down, they need to pass on that life, that spark of life, and that is the DNA. And so you need that DNA to be passed on from generation to generation. The term genetic has to do with generations, right? Your grandma passes on her DNA to your mother, and your mother passes on her DNA to yourself. And so it gets passed on. If you choose to have children, you're going to pass on your genetic material onto your biological children. Okay, the other thing is that it's the code to make the proteins in those individual organisms. And then those proteins go on to control the chemical reactions inside an organism and to build quite a bit of the structures. A good number of the structures of our body consists of proteins. Our muscles, the skeletons of our cells, the networks of our bones, the stretchiness of our skin, the color of our eyes, the color of our skin, the color of our hair, that is all protein. All right, the letters in DNA, we're not talking about the bases, but we are talking about like the literal what does DNA stand for. By the way, we are now talking about the DNA structure, what does it look like? And so the D stands for deoxyribo, the N stands for nucleic, and the A stands for acid. So it stands for deoxyribonucleic acid. And so that leads us to the next one, what type of macromolecules? If you watch the Amoeba Sisters video, it is biomolecules. And so therefore it is classified as a nucleic acid. And you can remember that just by the name. Nucleic acid is in the name DNA right there. I don't believe you're going to be tested on this one, and there is a typo. They did not win the Nobel Peace Prize. They won the Nobel Prize in, I believe, chemistry or biology. But regardless, they won a Nobel Prize for discovering the structure of DNA. The main two that you're always going to hear almost every time is Watson and Crick. But another gentleman named Wilkins also shared the prize with them. Had culture been a little bit more fair, and had the Nobel Prize been able to be awarded to individuals that are no longer alive, very likely Rosalind Franklin should have and would have won the award as well. Okay, DNA is described as a double helix. Really, the double helix comes from the fact that the spiral shape is twisted around itself twice. And so that's the main thing. It has a spiral shape. That's the helix portion of the name. But you can remember the double portion because it is double-stranded. Okay, DNA is a polymer. It is consisting of subunits or building blocks or monomers called nucleotides. Nucleotides are the big long chains that make up DNA. And they are made of 3 parts. The first part is the phosphate group. We've talked about this quite a bit. Phosphate group is usually shown as a circle. Sometimes it has a P for phosphorus in it. It'd be the only thing that has that phosphorus in it. And so usually we designate it or have it be shown as being a circle, a circular shape, and you can see that right there. I'm going to change my highlighter to different colors just so we can match them up with the colors in the picture. And then we have the deoxyribose sugar. The sugar is a 5-membered sugar, so it does make a pentagon shape. And so we always, typically in cartoons and visuals of the nucleotides, it will be a pentagon or have a pentagonal shape. And then the last thing is the nitrogen base or the nitrogenous base, depending on where you're reading that information from. And that is sticking off of the sugar. The sugar is the central portion of it. And there's different ways the bases kind of look. They do have nitrogens in them, so if you see the chemicals of it, it's a dead giveaway. We like to do them similar to rectangles. Sometimes those rectangles will have dimples or outputs or little like jut outs of them to show kind of how those bases connect, but they don't always have that. The phosphate is always going to be a circle though in most diagrams, and the sugar is going to be a pentagon. So the third one must be the base. Speaking of the bases, there are 4 major bases: adenine and thymine, or A and T, and they go together. Adenine and thymine go together. They complement each other. They say, hey, you're looking very nice today. No, they fit together based on the number of hydrogen bonds. A will always pair up with T unless there's a mutation, and G will always pair up with C unless there is a mutation. Guanine and cytosine are the last 2. All right, when we look at the molecule of DNA, you have the phosphate groups, which I circled in black. They run along the sides of the DNA molecule and the RNA molecule. It looks very similar to this. RNA does look very similar to this. So they run along the sides. And then I put squares around the nitrogen base pairs. Since it said pairs, I'm trying to make these as square shaped as possible. But those nitrogen bases are sticking out in the middle like rungs on a ladder. Okay, and then I did the pentagon in green. Okay, and then I put a blue rectangle around one nucleotide, but you can see that there are many nucleotides here. I can keep going, right? Phosphate sugar base. Phosphate sugar base is a nucleotide. Phosphate sugar base. The rungs of the ladder are all phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. We call that the backbone. And then the bases stick out from the sugars. Base, sugar, base, sugar, base, no treble. All right, and then the other side would be exactly the same, only it has a different direction. Remember, these things have directions based on the gizmo that we did, as well as other practice. Oops, and I made a mistake. But we have the opposite sequence. But notice the base is always attached to the sugars, and the phosphates are always kind of by themselves, and they make that ladder shape there. One nucleotide in my picture is the phosphate, the sugar, and the base. So there's a nucleotide that I just put an L around. Okay? All right. So in between the 2 strands of DNA, they are held together by what is known as a hydrogen bond. Hydrogen bonds are pretty strong. They're the strongest type of intermolecular force. You will learn about that in chemistry, whether you take general level chemistry or whether you take accelerated chemistry. But what they are is they're able to be opened up somewhat. They're able to separate pretty easily. They're not quite as strong as a true as a true covalent bond, but they are strong enough to hold the two strands together at the very least. But it allows it to open up during replication and transcription. So if you look at my hot pink marker, right there is where the strands separate during transcription and replication. Okay, the bonds that connect the phosphates to the sugars right here that I'm highlighting with my hot pink, those are covalent bonds. Those are super strong. You need a very strong chemical reaction and a lot of energy in order to break those bonds. And that's a good thing, because if those bonds get broken, right, then what happens is the code could get scrambled. And that leads to crazy mutations, and we don't want that to happen. So we want the hydrogen bonds in the middle so that the two strands can separate, but we don't want the bases to break out and be scrambled and replaced in the wrong sequence. Okay? So in the next one, we're going to use complementary base pairing rules, and I'm sorry I haven't been circling the numbers that I've been on. We did number 12, which is the bonds, and number 13 right there. All right, next one is the complementary strand for the DNA sequence. So all you're doing is remembering your complementary base pairing rules. A goes with T, G goes with C, A with T, G with C, 8 the graham cracker, 8 the gummy candy, apple tree, garage car, whatever helps you remember it. A goes with T, T goes with A, G goes with C, G goes with C, C goes with G, C goes with G, and so on and so forth. So the sequence of DNA, complementary strand of DNA, is TAC, CGG, CAT, ATG, GGT. All right, the next one is that is complementary base pairing rules. This is called Chargaff's rule. And what you need to know for this one is that C is going to equal G, and that T is going to equal A. All right, once you know those two things, if it contains 30% cytosine, we know that the guanine is going to be the same. We know that all 4 of them put together have to be 100%, because that means all of them. So if my C and my G is 60%, because 30 plus 30 is 60, that means that the remaining T and A have to be 40%. So we take that 40%, since they have to be equal, it's very similar to doing the 2x equals 40, and then you divide both sides by 2. It ends up having both of those being 20. And then you just double check. You make sure that the A is equal to the T, yep, yep. The C is equal to the G, yep, yep. And that they all add up to 100. And when you take 30 plus 30 is 60, plus 20 is 80, plus 20 is 100%. Okay, next one, we're going to move on to DNA replication. What organelle does DNA replication take place in? It is the nucleus. The steps involved in DNA replication, the enzymes unzip, the helicase unzips and separates the DNA. You're adding new bases by DNA polymerase, and then DNA ligase comes in and rebonds the backbones, and then the 2 new strands of DNA rezip. And you get 2 identical copies at the very end. As far as the code is concerned, the code is identical. So we're going to see that in the next one right here, right? My original code, I'm replicating this, is TAAT, CG, CG, GC, TA. Notice both of these have the same exact code. So my original strand is on the outside because they unzip from each other. So you separate here, and these 2 unzip, they move to separate sides, and then the new strands fill in on the inside right here. They fill in on the inside, and then polymerase or ligase comes in and rebonds everything, make sure that code stays in place. Okay, moving on to transcription. We're moving on to transcription. DNA transcription is when you make a molecule of mRNA. Why do we have to make a molecule of mRNA? We have to make it to get the code out of the nucleus. Right? The DNA is stuck inside the nucleus. My DNA is stuck here. My ribosome, where the protein is made, is outside the nucleus. DNA can't get through. Oh no, what's going to happen? Well, it has to make a molecule, and that molecule is the messenger RNA, which then can go into the ribosome. So it takes place inside the nucleus because you're copying the code, you're writing down the code, transcribing the code. It literally means to write down or to write across. So you're just copying down the same exact code. The product of DNA transcription is mRNA. You need to know the differences between DNA and mRNA. And any RNA. The number of strands of DNA is 2. The number of strands of RNA is 1. Type of sugar, DNA has deoxyribose, RNA has ribose. Type of bases, for DNA, thymine, adenine, cytosine, and guanine. The main difference is that in RNA, there is no T. Instead, you have a U, and that U stands for uracil. Okay? In this case, when you have RNA complementing DNA, don't forget that T becomes A for RNA, and that A becomes U for RNA. So when you're going from DNA to RNA, you always complement that way. Okay, next one, we're transcribing the following DNA molecule. So in the first step, we have the 2 strands of DNA separating. In the second step, we are adding the new nucleotides only to the first strand. Notice the second strand we are ignoring. mRNA is only 1 strand, so we only use the coding strand as a template, so we're only making 1 strand of DNA. At the very end, boom, you get the 2 strands of DNA coming back together, and you get the mRNA leaving the nucleus. The sequence of mRNA I got was UAU, GCC, UUU, AAU, GCU, and A. Okay, translation. The point of DNA translation is to code from the mRNA and to make the protein. It happens outside of the cell. The product of DNA translation is an amino acid sequence or a protein. Amino acid sequence is more scientifically correct, but we can call it a protein for this class at this level of biology. If you take a more advanced level, never call an amino acid sequence a protein, because what the difference is that the protein is all folded. Sometimes you cut off a couple of amino acids to make it nice and neat, and sometimes proteins pair up with other proteins or vitamins and minerals in the body in order to do their function. So a protein is like the final polished product with the final set coat of paint, whereas the amino acid sequence is like the basic underlying structure. So, but for this class, we're going to call them the same exact thing. Okay, the codon is a 3-base sequence found in mRNA. It's a coding sequence. It equals 1 amino acid. Every 3 bases or every 3 nucleotides in mRNA equals 1 amino acid. The anticodon complements the codon, and they are found in tRNA. So if my codon is, for example, UUG, right, my anticodon on my tRNA is going to be AAC. So they complement each other. U goes with A, U goes with A, G goes with C. Okay, the building blocks of proteins are amino acids. You have 20 different amino acids. There's something like 64 different codons, and so certain codons code for the same amino acid. Next one, we're going to translate the following molecule into DNA. So remember, you do not use the second strand. It is the non-coding strand, the nonsense strand, or the silent strand. Those are some of the names for it. There's others as well. The strand number 1 is the coding strand. And so what you do is you look at the letters and you complement it using RNA. So A becomes U, T becomes A becomes U, C becomes G, and so on and so forth. So I got this mRNA strand right here, UAU, GCC, UUU, AAU, GCC, and UAA. And then what you need to do is you need to take that mRNA strand and turn it into an amino acid strand, and you use that by the codon wheel. Okay, so I'm going to do one example. So we have UAU. That's my codon in mRNA. Notice I divided up my mRNA into codons, so I know how many I need to do. And so I go to the mRNA wheel, and I start in the middle, and I work my way out. U, A, U, and I get TYR, tyrosine. All right, and so that is TYR right there. And I do that with my next ones. I get alanine, I get phenylalanine, I get alanine again. And then the last one, UAA, I put an X here, but you don't actually have to put anything there. And the reason why is because when I look up UAA, start in the middle, UA, and then go to A, I see it's a stop codon, and that means that I am done. Okay, next one. There is a typo in this one. There should be arrows in between those molecules. We'll try to get that fixed for later. But when you go from DNA to mRNA, that is transcription. When you go from mRNA to protein, that is translation. This whole sequence here can be called protein synthesis, or your book more often refers to it as gene expression. So in other words, it's making the product, it's expressing the product of the gene, which is a piece of the DNA that codes for a protein. Okay, 32 mutations, pretty easy. Deletion mutations are when nucleotides or bases are lost in the sequence. So for example, here I have ATA, CGG, AAA. Here I have ATA, that's the same, but I only have the C. I deleted 2 of my G's. And so that's a deletion mutation. Just like when you delete letters in a letter, you're deleting letters in the sequence. Insertion is the opposite of deletion. In an insertion mutation, you are adding new nucleotides, adding extra nucleotides. And so in my example here, I put in a T where there is no T to begin with. In a substitution reaction, you replace the nucleotides. So you're keeping the same number of nucleotides. You're just removing one and replacing it with the wrong letter. So notice here I have ATA as my first 3 nucleotides. Here I've replaced the T and the A with an A and a G. So use your creativity, come up with a different mutation for each one. And yeah, all right. Last but not least is not every mutation is harmful. All right, and here's a couple of reasons why, and there are additional reasons why as well. Sometimes the protein stays the same, okay? So let's say I have a DNA molecule that codes for the mRNA UCU. So UCU is my mRNA, so UCU is my mRNA. That means my DNA would have been AGA. So my DNA is AGA. It makes the mRNA that is UCU, and that makes serine. Okay, so there we go. DNA to mRNA made serine. All right, now I'm going to mutate my DNA. I'm going to mutate my DNA, and I'm going to change it to, instead of UCU, I'm going to change it, to UCC, for example. All right, in order to do that, my DNA must have mutated to AGG. But look, AGG makes UCC, and that still makes serine. So the protein doesn't change at all, even if you have a mutation sometimes. Some of the mutations can change the protein, okay? But the protein still works as well. So the protein works just fine. It does its job. The organism doesn't change at all. All right? Other mutations can happen in an area that doesn't make protein. All right? So no protein gets changed. And then last but not least, some mutations can change the protein, but lead to either neutral or beneficial traits to the organism. So you can have a trait inside of your body that does nothing bad for you, or actually could do something good for you. So some examples, lactose intolerance. That is the normal default setting for human beings. If you can drink milk as an adult without getting sick, you are a mutant. That is a beneficial trait for humans. It's another source of food and energy. It can be neutral as well. We learned about red hair and blue eyes. For most people, that is a neutral adaptation. That's a neutral trait. It doesn't help the organism to have blue eyes. It doesn't hurt the organism to have blue eyes. It's neutral. A lot of these harmful or neutral or even beneficial traits are called variations. And so all mutations, all of our variations, all of the differences in between species and between individuals within a species, all of those originated as mutations. And so if all mutations were harmful, there would never be any evolution. There would never be any advantage to gaining new beneficial traits. There would be never any new beneficial traits coming along anyway. All right. So number one reason why is because sometimes nothing changes at all. And sometimes mutations can lead to either neutral changes or even beneficial changes. in those organisms. Okay, so I hope you paid attention. I hope you rewound, fast forwarded. I hope this was some benefit for you. I hope to see you guys for the test day. We did get everything squared away here, so I should be good to go. And I'll see you soon, Pirates. Have a great day.